Hello and welcome. Here is my brief introduction. Welcome back. We start with our first training in physiology, and today we'll be discussing cell membrane. There would be integration with pharmacology, pathology, obstetrics, genetics. So let's start. Cell membrane is covering of the cell, and it is comprising of proteins 50 to 55%, lipids 40 to 45%, and carbs 3 to 5%. There is an exception. That's myelinated nerve cell membrane. It is comprising of more of lipids, that is 75 to 80% as compared to 20 to 25% of proteins. The major functions of cell membrane: one, which is very obvious, yes, it acts as a covering. Second, it acts as a gate. Gate means so many substances will be coming and going into the cell, the thing what is called as the transport. Now if it is using energy, then we'll call it the active transport, and if it is going passively, obviously it would be called as the passive transport. In our second lecture, we'll be talking about this active and passive transport. The third major function is support. There are anchor proteins which gives good support to the entire cytoskeleton, and they are attached with the cell membrane. Now this cell membrane, it is made up of phosphate plus lipids combined, or the combination of both, which is called as the phospholipid molecule. Now this phospholipid molecule is amphiphilic. Amphi means both. Over here we are dealing with water and lipids, and philic means loves. So it loves water and lipids both. But over here we are collectively talking about the whole of the cell membrane, or whole of the phospholipid molecule. If we see really in detail, now this phospholipid molecule, it has got two parts. One what is called as the head part, and second what is called as the tail part. This glycerol is a junction between both of them. Now this head part, it is negatively charged because of this phosphate group, so we'll call it as polar. And this part, that is the head part, is hydrophilic. The tail part, which is a fatty acid, it is lipophilic, but at the same time it is hydrophobic also. So thus cell membrane has got two layers of phospholipid, so that's why it is called as the phospholipid bilayer. Now as we said that this tail section, it is hydrophobic, so that's why there won't be any water in between. So if we see as a broader aspect over here, say this is the cell membrane. Inside the cell, it is called as the intracellular fluid, which is an aqueous environment, and extracellular fluid, which is outside the cell, that is also an aqueous environment. So it is the cell membrane which is keeping both of them separate. Now there are few proteins which are present on the outer membrane, and there are few proteins which are present in the inner membrane. They both are different. Similarly, you will find carbohydrates outside and very less carbohydrate onto the inside of the cell membrane. Now when we look at all these things together, that is first point, that is outer and inner phospholipid layers, they are different. There are different peripheral proteins, that is proteins which are on inner and the outer membrane, And carbohydrates, they are present on outer membrane. There are very less or almost none on the inner layer. So this tells that there is a clear difference between outer membrane and the inner membrane, and that's what is called as the membrane asymmetry. This is what is called as membrane asymmetry. Now this takes us to what's called as the selective permeability. Permeability means ability to pass through that membrane, but it is selective. It depends upon one, polarity, that means whether the substance is charged or it is neutral. Then it depends upon the size of the molecule, whether it is a small or large. So if we really see it in this way, that when we have got a non-polar and a small molecule like oxygen and carbon dioxide, so they will be passing very smoothly and rapidly across the membrane. Against that, when we have got polar, that means charged, and the small molecule like water, so they will be passing through very slowly. When we have got large molecule, but at the same time they are non-polar, so yes, they will also pass, but again they will also pass quite slowly. Finally, large molecule and the charged like glucose, they won't pass directly. And if they are highly polar like say sodium or potassium chloride, they don't have any chance to pass through it. Lipid solubility, that is also very important. Remember, lipid soluble, they can pass through. We'll be taking all these points into our discussion as we proceed. So over here, so there is extracellular fluid and there is cytoplasm, which is acting as an intracellular fluid, and they both are separated by phospholipid bilayer. And there are carbohydrates, glycoproteins, there are peripheral proteins, cholesterols, etc. more as we proceed. But this entire concept was called as the fluid mosaic model. This concept was given by S.J. Stinger and G.L. Nicholson in 1972. Experimental evidences are like labeling experiment. Over here, using spin labels. Spin labels, they are like organic molecule. They have unpaired electron ability to bind and they are usually nitrogen. So they are used as a tool for probing proteins. And then using electron paramagnetic resonance, it is evaluated. Similarly, we have got the X-ray diffraction, where we'll be watching the diffraction of the incident X-ray beam due to specific substance, or it is also called as the X-ray crystallography. The third is calorimetry. So over here, the result is that there is integral membrane protein, they diffuse at different rates, and that is affected by viscosity of the lipid bilayer. Molecules within the cell membrane, they are dynamic. They slide and glide. So these were the concepts which were given by Stinger and Nicholson. From this point onwards, we'll be proceeding like this brain map. This is cell membrane, which is our today's topic. Now this cell membrane is comprising of lipids, proteins and carbohydrates. That's we talked about. And lipids, they are divided into phospholipids, glycolipids and cholesterol. And then as we'll proceed, we'll keep on taking up that okay, this topic has been done, and thus we'll complete the whole circle. Let's start. So moving on to the lipids, first we'll be talking about phospholipids. So in lipids, when we say phospholipids, that means there is a phosphate group which is present, and this is highly abundant. Outer membrane it has got phosphatidylcholine. It's also called as the lecithin. This is a major surfactant. When we say surfactant, that means it is surface active agent. They lower the surface tension between two substances. So when we talk about pulmonary surfactant, this pulmonary surfactant, it is formed by type 2 valvular cells. And what does they do? They facilitate breathing by increase total lung capacity. At the same time, they keep lungs inflated. Now we are dealing with right now outer membrane, right? We are dealing with outer membrane. First one was phosphatidylcholine. The second substance is sphingomyelin. That is found in membranous myelin sheath surrounding some nerve cell axons. So its role is insulator of nerve fibers, and its importance is that evidence shows presence of sphingomyelin in inner membrane also. This is very important. The reason behind it is that sphingomyelin, it gets converted to ceramide by the enzyme sphingomyelinase 2. Now this is exclusively in inner membrane, and this ceramide is involved in apoptotic signaling pathway. And this apoptotic signaling pathway, apoptotic is programmed cell death. So how it really occurs? We'll see it. Meantime, sphingomyelin, when it accumulates in hereditary disease called Niemann-Pick's disease, this is Niemann-Pick disease. There are two types, type A and type B. Type A, it occurs in infants. So they'll be having jaundice, hepatomegaly, extensive brain damage. While when it occurs in preteens, there would be hepatosplenomegaly, but brain is not affected. That's the difference between both of them. Now another important point is that in obstetrics, for fetal lung maturity, this LS ratio is used, lecithin sphingomyelin. So if it's more than 2, fetal lung maturity is speculated. However, its sensitivity is low because false negative results, they are frequent. So amniotic fluid, which has got lecithin and sphingomyelin, they are put it into test tube, they are shaken. If there is formation of bubbles, so then it is considered as positive. This is also called as the bubble test or shake test, or it's also called as the Clement's test. Clement was the German scientist who gave this concept. So we talked about phospholipids. It's outer membrane, that is outer membrane of cell membrane. Now we talk about the inner membrane. This inner membrane, it has got phosphatidylserine. This phosphatidylserine, its function is cell signaling for apoptosis. So when this phosphatidylserine, which is into the inner cell membrane, as it appears on the outer side, it is a signal for macrophages to engulf. It has got a role into coagulation also. So when there is site of injury, platelets, as they encounter this site of injury, they see this, then these phosphatidylserine, it will come out. It will act as a procoagulant surface. On this procoagulant surface, this would be the surface for tissue factor, factor 7, plus there will be activation of factor 10, and all these things, they will lead to formation of thrombin. Now in case if anything goes wrong to this stage, right, as this photo cut, as this phosphatidylserine, in case if it is not going out, so if this process is disrupted, That's what is called as the Scott's disease. And in Scott's disease, as you must have guessed, that if it is not acting as a procoagulant surface, there will be problem in the thrombin formation. Obviously, that will lead to bleeding disorder. The dietary sources for non-veg, meat and fish, and for veg, white beans and soy. Some amount, in fact it's a very small amount, which is present into dairy products. Annexin is a substance, in fact annexin A5. This is cellular protein in annexin group. So it binds with phosphatidylserine, and when it is expressed on outer membrane. Now if it is expressed on outer membrane, it makes a complex with annexin A5. So this thing is a marker for apoptosis, and it can be detected by flow cytometry. So humans have annexin A 1, 2, 3, 4, 5, 6, 7, 8, 8, L1, 8, L2, 9, 10, 11 and 13. The fourth is, we are talking about inner membrane. And in inner membrane, this is the second, as such the fourth. Phosphatidyl ethanolamines. This is found in all living cells, particularly in white matter of brain, nerves, neural tissues and spinal cord. Now its importance is to catch infectious prions. These prions, they are nothing but they are the misfolded proteins. So it is necessary to remove these misfolded proteins. Also, the importance of phosphatidyl ethanolamine is its role in secretion of lipoproteins in liver. It also helps in coagulation, helping phosphatidylserine by promoting and binding to factor 5 and factor 10. So thus we talked about phospholipids, external membrane, we talked about internal membrane. Now we'll be going for few other phospholipids. That would be inositol triphosphate and cardiolipin. So our fifth candidate is IP3, inositol triphosphate. It's also called as the INS3P or inositol 1,4,5 triphosphate. Now the story inside the cell. PIP2, phosphatidyl inositol 4,5 biphosphate, which is in plasma membrane. It is hydrolyzed into IP3. Hydrolysis is done by phospholipase C, so this is an enzyme. Now this IP3, it acts as a second messenger, that is along with IP3. It works along with DAG, diacyl glycerol, and it has got signal induction in biological cells. How it occurs? Say DAG, it stays inside the membrane only, but IP3 is soluble, so it diffuses. It diffuses and it acts on IP3 binding receptors. They are on endoplasmic reticulum. Now these are in fact calcium channels. So in presence of IP3, they open up and calcium is released into cytosol. Now this calcium which is released into cytosol, that will stimulate intracellular signals which are calcium regulated. Say for example in smooth muscles, when calcium concentration rise, it leads to contraction of muscle cells. The next one is cardiolipin, also called as Calcutta antigen. It is component of inner mitochondria membrane. Now the name cardiolipin first found in animal heart. It was first found in animal heart, and it was a beef heart in 1942. That was done by Pangborn M. So he published in Journal of Biology isolation and purification of a serological active phospholipid from beef heart. That was the article. Now this is present in mammalian cells. It's also in plant cells but exclusively in inner mitochondrial membrane. The function is it regulates aggregate structures. At the same time, it triggers apoptosis. Apoptosis, it is programmed cell death. So how it occurs? This is onto the inside of the mitochondrial membrane. So when this cardiolipin it goes out, when it comes out, there is release of cytochrome C. This cytochrome C, it binds to IP3 receptors on endoplasmic reticulum. Obviously these are the calcium channels as we discussed. So there is release of calcium. When this calcium is released, this calcium will go to further release of cytochrome C, and the whole cycle is established. This is what is called as the vicious cycle. Calcium level keeps on rising, and when it reaches to the toxic level, it leads to cell death. Other functions, they import protein into mitochondrial matrix, and there is an anticoagulant function also. Now there is a clinical importance of it. Say cardiolipin, it is necessary that it should be biosynthesized. So for biosynthesis of cardiolipin, there is an enzyme called tafazine. This tafazine is needed. But there is a syndrome called Barth syndrome. So in Barth syndrome, there is mutation of a gene which is coding for tafazine. So if tafazine is affected, that means cardiolipin won't be created properly. It won't be biosynthesized properly. So result, there would be abnormal mitochondria, and that would lead to cardiomyopathy and general weakness. In Parkinson's disease or in Alzheimer's disease, Parkinson, so it affects speed of thinking, memory, problem solving. In case of Alzheimer's, it affects language and memory. But over here, in both these cases, it's a mitochondria, either because of age or because of free radicals. There is lipid peroxidation. Now when it occurs in substantia nigra, right, it leads to Parkinson's disease. And in early stages of Alzheimer's also, this thing is seen. But in both the cases, cardiolipin level is definitely dropped. Similarly, in other diseases like non-alcoholic fatty liver disease and heart failure, there is decreased level of cardiolipin. In early stages of diabetes, there is decreased level of cardiolipin. Now in case of syphilis, which is by Treponema pallidum, there is a test called Wassermann test. Wassermann test for syphilis. Now in this, cardiolipin from cow heart, it is used as antigen. And then we have got anti-cardiolipin antibodies. But the thing is that they can increase in even other conditions also, like SLE, malaria, tuberculosis. So in all these conditions, you will find rise in anti-cardiolipin antibodies. That's the reason that newer tests like RPR, that is Rapid Plasma Reagent Test, or VDRL, Venereal Disease Research Laboratory Test, they are used for screening of syphilis. They are used for screening of syphilis. So we talked about phospholipids. Now we move on to the second component, that is glycolipids. So the way we were having, say, phosphate group for phospholipids, we have got glycolipids, in which there is combination of carbohydrate plus lipids, and they are attached by glycosidic bond, and this is a covalent bond. So main role is stability of cell membrane, and they facilitate cellular recognition. This is very crucial for immune response. Now if you see these glycolipids, they are two types. Glyceroglycolipids. These glyceroglycolipids, they are for photosynthetic membrane. Right? So they are of importance to plants. Glycosphingolipids, they are cerebrosides, gangliosides, globosides, glycophosphatidyl inositols. But out of all of them, these first two, cerebroside and gangliosides, they are found in brain. It's also present in intestine. Now see, I'm writing GM1. GM-1, I have put a cross on it. We'll see why. So this GM1, which is a prototype ganglioside, 1 because it is with one sialic acid residue. Now its function is neuronal plasticity and the repair mechanism. Now see what happens in intestine. Whenever there is Vibrio cholera microorganisms, now they produce cholera toxin. Now this cholera toxin is very specific. It's also called as the AB5. The reason behind it is that it is a complex with 6 protein subunits. And these 6 protein subunits, that is type A. This type A, it activates, so there is an enzymatic function. There is another group which is called as the B subunit. And there are such 5. So it binds to GM1 ganglioside on surface of target cells. Now when they bind on the surface, they form what's called as the toxin complex. Now this toxin complex, it is endocytosed by cells. Once it is inside the cell, it is the A or the activator, it comes into action. So CTA1, it is released in cell. That would activate GS alpha subunit. Right? This is GS alpha subunit. Now this is stimulatory, so it will increase adenyl cyclase activity. Now we know that as adenyl cyclase activity is increased, straight away it will lead to increase in cyclic AMP. Now this value rises so much, it is over 100 times the normal value. Now because of increased cyclic AMP, there is stimulation of cytosolic protein kinase A, the very vital component. And this active pKa or the active protein kinase A would act on CFTR. This is nothing but it is simply a chloride channel protein. It is a chloride channel protein. Now the CFTR or the cystic fibrosis transmembrane conductance regulator, as it is stimulated, there would be ATP mediated efflux of chloride ions. There would be efflux of chloride ions. Along with that, there would be secretions of water, sodium, potassium, bicarbs. Result, there would be rapid fluid loss, over 2 liters per hour, and that's what is called as the rice water stool. Now very similar mechanism is there for pertussis toxin. In that case also it is AB5. It is by Bordetella pertussis, and that's what leads to whooping cough. The only difference is that over here it is the GI alpha subunit is affected. So due to this, because it is inhibitory, and as it is affected, so it would be unable to inhibit cyclic AMP production. So there would be continuous action of cyclic AMP because it is unable to inhibit cyclic AMP production. So there will be overreactivity of cyclic AMP. This B subunit, it is quite non-toxic and can be bind, and it is routinely used as a neuronal tracer. Okay, we were talking about that GM1 which is not equal to GM-1. GM1, to now we know that it is nothing but ganglioside. GM1 in fact is a disease. It is a gangliosidosis 1. It's a progressive neurological genetic disorder. So abnormal accumulation of lipid in cells, especially brain cells, is what really happens in gangliosidosis. Affected children, unfortunately, they don't cross even 2 years. So we talked about phospholipids, we talked about glycolipids. Now is the turn for cholesterol. And we are still talking about lipids. And as our main topic is cell membrane. Okay. So cholesterol, which is third type of lipid, its main function is to maintain fluidity. Keep this thing in mind, it maintains fluidity. Higher fluidity of membrane, it is much better for health. So when there are unsaturated fatty acids, right, they are very loosely bound. There is space in between. While it comes to saturated fatty acids, they are quite tightly bound. So unsaturated fatty acids are also called as the PUFA, polyunsaturated fatty acids. And then there are essential fatty acids. All these are very good for the health because they increase fluidity. Increase in fluidity is good for the cell membrane. So these are like linoleic acid, linolenic acid, and arachidonic acid. These three acids, they are very good for health. The dietary source is olive oil, peanut butter, almonds, cashews, sesame seeds, avocados, right? All these foods, they are well rich in unsaturated and essential fatty acids. Coming to saturated fatty acids, they are not good for health. Palmitic acid, stearic acid. Do remember the names. Now this is a concept, transition temperature. Transition temperature is that temperature when the solid lipid, it gets converted to fluid lipid. And this is a very important point to remember. Transition temperature is inversely proportionate to fluidity. So that means in case of unsaturated fatty acid, there is lower transition temperature, thus higher fluidity. We talked about unsaturated fatty acid loosely bound, so that's why there is higher fluidity. The reverse in case of saturated fatty acid, higher transition temperature, so therefore lower fluidity. Lower fluidity means it is not good for health. The function of cholesterol, it maintains fluidity, we talked about, and this is a new word, that is what is called as the fluidity buffer. So characteristics of lipids: 1, fluidity buffer; 2, membrane asymmetry. Now we know that why we called it as membrane asymmetry, because different phospholipids, outer layer, inner layer, different proteins, outer layer, inner layer, carbohydrate present outside, negative inside. So that's why this entire thing is called as the membrane asymmetry. And the third function is repair. So post injury or exocytosis, endocytosis, any injury, it repairs it. Right? So that's, we are talking about cholesterol. Okay. So we talked about lipids fully. Now is the turn for protein. We'll be considering transmembrane protein. Transmembranes mean this is the cell membrane, and something which is through and through this membrane is called as the transmembrane. Peripheral, it means it is either here or it is either here, outside. it's called as the peripheral protein. Finally, there would be carbohydrates. So moving on to the protein part, whenever there is cell-to-cell junction, so there are two types of junction. The first junction, which is not very tight, right? It is like a velcro, and they are called as the cadherines. In case if there are tight junctions, so they are like as if we have put the rivets, and they are waterproof. Now from the transmembrane protein, they are also called as the integral protein. It means you can't really separate them from the cell wall or the cell membrane. If you try to do that, it will completely disrupt the membrane. The first one is something like channel. Say for example, aquaporine channel. They simply act as tunnel. Now water, very less water actually traverses. But over here, when it is channel aquaporine, so that means it will allow passage of water easily. The second one, it is called as the carrier, or they are the transporters. Say for example, glucose transporter 4. Now over here, these carriers, they will transfer the specific substance from extracellular fluid to intracellular fluid, or vice versa. The third type of protein is enzyme linked protein. Now there are few proteins where the enzyme would be necessary, and then they start functioning. Now these are like sodium potassium ATPase pump. So even against gradient, even they move from high concentration to low concentration, but they can move from low concentration to high concentration also. And finally, we have got the receptors. These receptors, which are GPCR, they are called as the G protein coupled receptor, also called as the 7-pass receptor or serpentine receptor. Very simple logic. They cross the membrane 7 times. So 1, 2, 3, 4, 5, 6, 7. Yes, it crossed 7 times. So that's why it is called as the 7-pass receptors. Now this receptor, this topic is so extensive. We'll be having one specific session only for receptors. But what they really do, that as soon as a specific substance, it comes over here, it will send a response signal at its tail end. So over here, this particular receptor or 7-pass receptor or G protein coupled receptor, right? This substance, which is called as the ligand, it could be chemical, hormone, drug, neurotransmitter, anything. But when they act over here, you will see some response at the tail end. And then there are anchored proteins. These anchored proteins, they are for support to cell structure, so that cell is not collapsed or broken. So there is lipid which is attached, and it is IP3. So protein is called GPI, glycerol phosphatidyl ionositol. So lipid attached is IP3. Now this is abundant in RBC, and that's the reason it prevents hemolysis. Let's talk about peripheral proteins. These are the proteins which will be either present on the outer membrane or the inner membrane. So for the peripheral proteins, because they'll be having supportive function, they are present in skeletal muscle and red blood cell. In case of skeletal muscle, it is called as the dystrophin. So if dystrophin is muted, skeletal muscle paralysis occurs, even respiratory muscles, because dystrophin is something which is crucial for the support of muscle. This is what is called as the DMD, or the Duchenne muscular dystrophy, in which there is progressive muscular weakness, difficulty in standing, even gait abnormality, gait is affected. And this inability to stand up is called as the Gower sign. Treatment, at least at this point of time, though there is none. CRISPR research, they might lead to cure for DMD. CRISPR is a very fine genome editing technology, and CRISPR stands for this clustered, regularly interspaced, short palindromic repeats. This is from the perspective of genome editing. Now RBCs, we saw that for the skeletal muscle there is dystrophy. For the RBC, red blood cells, we have got spectrine and ankyrin. So this spectrine and ankyrin, they both are vital for the biconcave shape of red blood cell. So they both are important. Spectrine, it acts as a cytoskeletal protein. So it lines intracellular side of the plasma membrane. Intracellular side. It is pentagonal or hexagonal arrangements. So something like this. Something like this. When there is mutation of spectrine, it leads to hereditary elliptocytosis, and that would lead to hemolytic anemia. So if you really see the appearance under microscope, this is it looks like. These are the shapes of RBCs. Regarding this ankyrin, it is integral membrane protein, but it provides binding sites for beta subunit of spectrine. So mutation of ankyrin, that is what leads to hereditary spherocytosis, and these spherocytosis, they lead to hemolytic anemia. Next is this DAF. It is a new name, decay accelerating factor, also known as the CD55 protein. Now this DAF is GPI anchored protein. GPI anchored protein. Anchored means it would be giving support. So in RBC, when there is low level of DAF and CD55, that means we are short of CD55 and CD59. Right? CD55 and CD59. They lead to complement mediated hemolysis because C5 and C9, they are called as the membrane attacking complex. So when they attack, it leads to hemolysis. That's the result, PNH, paroxysmal nocturnal hemoglobinuria. Symptoms, anemia, obviously because of hemolysis, fatigue, and then there are episodes of dark urine. Now the treatment is eculizumab, the drug which is acting against membrane attacking complex, the drug which is attacking against membrane attacking complex. Now this membrane attacking complex, right, which was say C5 to C9, right, that was the, yes, over here C5 to C9. How it acts? This eculizumab, it is a monoclonal antibody and it binds to C5. So now C5 cannot cleavage, it cannot break to C5a and C5b. Now this is good for knowing But eculizumab, it's by Alexion under the name of Soliris. This is the cost. Yes, I have not put any extra zero by mistake. It is $500,000 per patient per year. In India, it is available via restricted access program, but still it is that high. The cost is so high. This is how the dose it is given: IV intravenously 600 mg per week for 4 weeks. Then for the 5th week, that is after the 7th day, you'll be giving it 900 mg. And then every 2 weeks, you keep on repeating another 900 mg. So eculizumab, it is used for PNH, paroxysmal nocturnal hemoglobinuria, but it is also used for atypical hemolytic uremic syndrome. It is good to know. Atypical hemolytic uremic syndrome. Dose remains almost same, but higher doses will be needed over here. That is in the 5th and later on. So it would be 1200 mg per week. Now if we talk about that eculizumab is so costly, right? 5 lakh or $500,000 per patient per year. Here is the world's most expensive medicine, Zolgensma. The treatment would cost whooping $2.1 million US. There has been so much of criticism because of this astronomical cost, but well, it is said to have very effective for the treatment of spinal muscular dystrophy. So it's fine, just good to know. Now flow cytometry is used for diagnosis, because see, in case of normal cells, they will be able to reach sufficient quantity of CD59 and CD55. Now if they are deficient, so these are the GPI deficient cells. So here is, we talked about lipids, we talked about proteins, and now we talk about carbohydrates. Carbohydrates, which are 3 to 5%, important for survival. Now say this is RBC and there is Hutch antigen. Hutch antigen is nothing but a protein, and on that if there is NAG, NAG is what? N-acetyl galactosamine. That's nothing but carbohydrate, and we call it as blood group A. Over here, this is RBC, Hutch antigen, and you have got galactose. If there is galactose, so then we are dealing with blood group B. Now if both are present, we'll call it AB. If none is present, we'll call it O. That was easy. Now this was presented by Karl Landsteiner. He was Austrian and he got a Nobel Prize in 1930 for this. Now if there is no H antigen, that's H antigen. If there is no H antigen, we call it a rare blood group, that is Bombay blood group. But it's not so that only the Bombay blood group is rare. Some rare blood groups are like, say, golden blood. In case if there is no RH, it is called as the RH null. Complete absence of any of the RH antigen. Now this was discovered about 50 years back, and today it is considered that only about 50 individuals, they are known to have RH null or the golden blood. Now in 1990, something very interesting happened. Australian Red Cross Blood Service, a person came called SARA. She came as blood donor. Now as they were checking her blood, and they suspected a new antigen, and yes, then it was found So they named it as SARA. Nothing happened for 20 years. After 20 years, Canadian Blood Service, they came up against one case. A Canadian boy who needed massive blood transfusion at birth. Massive blood transfusion. Everything else was ruled out. So they suspected that is it the SARA blood group? So they sent the blood to Australian organization. And guess what? The test result was mother was SARA negative, that is with anti-SARA antibodies, and that kid, that boy, was SARA positive. So actually, anti-SARA antibodies of mother, they were attacking SARA positive cells. So then SARA antigen, now it is officially recognized by the International Society for Blood Transfusion. Now currently there are only 2 families in the world who are known to have it. Alright, so this was about the cell membrane. Thank you so much and see you soon.